Okay, so now let's turn our attention to the properties of acid production by the stomach. So that we know that acid production serves several functions in the stomach, and one of the primary functions it serves is an antimicrobial function. And the way it serves this is because the pH is going to be less than 2.0. So remember, neutral pH is considered at approximately 7.0. Our blood pH is approximately 7.5 in the blood. And the stomach will drop the pH down below 2, actually down to about 1.8. And this is due to the acidification by the secretion of hydrochloric acid. So the stomach is going to produce hydrochloric acid, and the cells that actually produce this are called the parietal cells. Now let's look at acid production, acid output based on a meal. So if we look at time, if this is time 0 and this is time after food consumption, and here we are measuring on the y-axis acid output, we will see that acid production in the stomach rises very rapidly following the beginning of a meal at time 0, and then slowly dissipates back down to closer to neutral pH. pH actually is about 6 when we consider down the basal state. And we're talking here on the order of 1, 2, and 3 hours. So there's a rapid acidification in the lumen of the stomach due to the consumption of food. So what we're going to take a look at next, first, is how is hydrochloric acid produced, and then we'll look at the regulation of this. Now the first thing we need to consider is how the parietal cell actually produces hydrochloric acid. So if this is the parietal cell and up here is the lumen of the stomach, the interior compartment of the stomach where digestion will occur, we have to generate hydrochloric acid. So the question is, how are we going to do that? Well, remember from our information we discussed in salivary gland, we talked about the combination of carbon dioxide plus water using the enzyme carbonic anhydrase. And this enzyme is going to produce bicarbonate ion plus a proton. And here we have the dissociation of carbonic acid to give our proton, which is going to ultimately decrease the pH. Now again, remember the CO2 is coming from metabolism. We'll talk a lot about this when we get into the oxidation of fuel sources. The carbonic anhydrase enzyme, interestingly, is one of the fastest enzymes known. It can convert approximately 10 to the 8th molecules of CO2 into bicarbonate per second. So this is a very rapid process. Now, the bicarbonate itself is going to be transported out of the cell on the basal surface. This is the surface side of the cell that faces the interior of the body, opposite of the lumen. Now when this happens, we are able to also exchange for chloride ion, chloride coming from the blood. This transporter is coupled between the bicarbonate export and the chloride import, and this is considered an antiporter. The transport of chloride into the cell is obligate on the export of bicarb. What that means is the bicarb must bind to the interior of the protein, chloride must bind to the exterior of the protein, and only under those conditions will there be a flip in this transporter to shuttle these ions across the membrane. Now we have our chloride being introduced into the cell. We have our proton that is being generated from carbonic anhydrase, and now we have the components in order to generate hydrochloric acid. But we next need to get this proton outside of the cell and into the lumen. This needs to be a rapid and efficient process, and so we are going to place a transporter on the apical surface facing the lumen, and these protons now are going to be transported into the lumen of the stomach. This transporter is also an antiporter, which means that it has an obligate ion that is going to be transported into the cytosol of the parietal cell. And this ion that's going to be transported is potassium. The important feature of this is that we are maintaining, as best we can, electrical neutrality. So we are transporting a proton with a positive 1 charge, a potassium ion with a positive 1 charge to keep neutrality. Now, the importance of this, to get these protons into the lumen to acidify the lumen of the stomach, is so important that we are not going to just simply rely on a diffusible gradient. So as we are generating protons under this reaction, we are increasing the concentration of proton here, lower concentration here, so we have a gradient that could potentially drive this process. We are going to couple this with ATP hydrolysis. So this transporter is a potassium proton ATPase transporter. So that means we are going to couple the ATP conversion to ADP, the hydrolysis of ATP, to drive this process. Now, technically, we could have so much potassium come in here inside the cell that we could block the function of this transporter because of that gradient. So what we're going to do is put a potassium channel. This channel is not a gated channel. It is specific for potassium, and it responds to the gradient of potassium. So as potassium concentration gets higher inside the cytosol, that will drive the export of potassium through this single pass transporter. Now, we have the H, the hydrogen, the proton of hydrochloric acid. Now, how do we get the chloride? Well, we have our trusty chloride channel. And this chloride channel is going to operate on a gradient. So if we have high concentrations of chloride in the cytosol, which we do from this antiporter being driven by the continued synthesis of bicarbonate ion, then this chloride can now exit the cell. So we have a concentration gradient of high chloride inside, low chloride in the lumen. The other thing that's going to drive this reaction is charge. Because this chloride ion is negatively charged, we have a high concentration of protons being pumped out into the lumen because of this ATPase activity. This positive charge will help draw the negative charge. So we have an electrical gradient as well as a chemical gradient